Okay, so one of the major things that's if you want to do single cell DNA or single cell DNA plus single cell protein, which is DAPSeq data analysis, one of the major provider for the genomics purpose is Mission Bio. So they have a tapestry platform which is used to generate single cell DNA plus single cell um, protein data. So after generating data, when you get the analysis file, there is they have facilitated a algorithm to analyze this, and which need to be essentially installed using the Anaconda. So the very first step that we will be doing in here is to activate our Anaconda and install it. So let's Mission Bio tapestry. So the algorithm they provided is Mosaic. So we search for the API and we see this is the Mosaic version 3.101. So then we go to the GitHub and we can see the developer has wrote some installation guide, and you need Conda to install it. They have developed it like this. So to begin with, let's very first we activate our Conda environments. So cd slash slash dr.ds Conda 3 slash d slash then you go to the source activate. Okay, just a moment. Sorry. There's a dot, it's a silly mistake. Then I go to the source activate. So base environment we have activated. Now we check the Conda environment list. So we have two environments. One is Anaconda 3, another is Anaconda environments Mosaic. So let's activate the Mosaic. So we'll type Conda activate Mosaic. So we have activated the environment. Then what does this say? This is what you have. So installation guide. Let's go through this. Install. First you install the version of the device. Okay. Ensure the Lambda is being used. So install. Okay. So let's try with this. Let's see it. So we need the Libamba solver, write it strictly, and others. So let's install the Libamba. Then we just try. Let it install. As you can see, all things will be installed within our specific. Let's set it up as they say. And the next is we need the solver also. So we install the solver also. Done. Now, for successful state, Anaconda, open your console and run the following command. Conduct create a Mosaic mission by Mosaic. So they have a certain let's check if they have Anaconda directly environment. So let's try Anaconda package. So we go in here, try to install it from here. Let's copy. If it does anything, it doesn't. So the next step, what we try to do is that we follow the command from here, and that is this one. And the following command. Okay, let me run it. Let's see if it works. We have the environment in there. That's not helping. So trying here is create a new environment. Let me see if it's installed in there. Okay, and activate Mosaic 1. Okay, now if I want to return notebook, what will happen in there? Having a return notebook. So to distinguish in between of these, what I will do, I'll run this Jupyter notebook in a separate port to avoid any conflict with my existing packages. So what I do, click on a new notebook. No. So to create okay, create a new kernel. So let me start. I think I need to create a new kernel in here. So let's create a new kernel. Okay, so what will it do? We'll create a new kernel in here, and to do so, what we'll do, Jupyter notebook port 8889. Still don't have any kernel, so we need to install the kernel. To install the kernel, we need this. Let's install kernel in here. Python m rpy kernel install. Name and name. Python. Mosaic. Okay, let's install the IP kernel. So install the kernel install IPY kernel. Okay, now run the same command as we did before. What's the problem now? Okay, let's try to restart it. Could be the reason. So what do you do first? Type on the bin source activate panda list conda activate mosaic 1 Python. I had it before, so there's no hint of so let's double check again. There was a typo in here, which are extremely sorry for, and it is null. So kernel in Anaconda is established. So now let's open it up. The Jupyter notebook we used earlier. And we specifically ask it to open it with that. And when you want to create a notebook, now we have Conda Mosaic. Conda list. You can see the packages in here. What do we have? The main thing in here is to check out the Mission Bio package installed or not. It's lovely installed. Thank you. So this was our whole presentation, and I hope this will help you. And as an output, next time when you want to open this environment, let me close everything and restart again. Let me just close the things. So how do you start it if you want from next time? So at the very beginning, what we do, we go to that specific environment in Conda. So we go to the specific folder. That folder is a directory, the cd home.txt and Conda 3 pin. Now in there we write source activate. We want this base environment to be activated, and then we go to the Conda environment list, and we have Mosaic 1. So Conda activate Mosaic 1. So it's activated. Now we ask the Jupyter notebook to open this specific kernel with a specific port so that we don't have mingling with others. So you have it now, and in here we want the notebook. We just open it like this, and we ask it to open with a specific. Done, and we run whatever we want. Conda list. This is the way successfully to install the Mosaic as well as the isolated environment creation as and Jupyter notebook opening. You see the mission values installed. That's great. Thank you for following.